Okay, so this could be host defenses part one. This is what we typically call innate immunity. So this week's worth of chapters is really getting into our immune system with host defenses part one and part two. So when we look at host defenses, there are three lines of defense. This chapter will cover the first and second, and chapter 15 will cover the third line of defense. In the first line of defense, this is your barriers that will block invasion at the portal of entry. This does not involve recognition of foreign substances. It's just a sort of a blanket stop gap, and it's very general in action. With the second line of defense, this is non-specific as well. It's internalized system of protective cells, and inflammation and phagocytosis also fall under this line of defense. And again, these two specific, the first and second lines of defense, are part of what we call innate immunity. The third line of defense, which we'll cover in chapter 15, this is what we refer to as adaptive immunity. This is acquired on an individual basis as each foreign substance is encountered by lymphocytes. Reaction with each microbe will produce unique protective substances and cells that can come into play if the microbe is encountered again. And this provides long-term immunity. So again, here we can see a little descriptive looking at the three lines of defense. Again, first line of defense, this is surface protection that's composed of anatomical and physiological barriers that keep microbes from penetrating sterile body compartments. This is your physical barriers. It also includes your microbiome, as well as any chemical barriers. The second line of defense of the innate immune system, this is a cellular and chemical system that comes immediately into play if infectious agents make it past the surface defenses. And this will include cells such as phagocytosis, cells involved in inflammation, fever, and antimicrobial products. The third line of defense, which is part of your acquired or adaptive immune system, this includes specific host defenses that must be developed uniquely for each microbe through the action of specialized white blood cells. And these specialized white blood cells will include your lymphocytes, which are your B cells and your T cells. Now, a healthy functional immune system is responsible for the surveillance of the body, recognition of foreign materials, as well as the destruction of entities that are deemed to be foreign. And what these immune cells will be able to do is they will be able to contact with self cells, and you should get no reaction with your immune cells and your self cells. But if you have contact with a foreign cell, the detection and recognition of a foreign cell or virus, and this should lead to the destruction of that foreign cell and or virus. Now, white blood cells move throughout the body, searching for potential pathogens. These white blood cells are trained to recognize self, which are your cells, or body cells, and non-self, bacterial cells, or other invading pathogens. Markers, these are molecules that are found on the surfaces of cells. They are typically composed of proteins and or sugars, and they are evaluated by cells of the immune system. Now, the tissues, organs, and cells that participate in immunity. We have mononuclear phagocyte system, which will include cells like your macrophages and your neutrophils. Spaces surrounding tissues that contain extracellular fluid, the bloodstream, and the lymphatic system. Now, the lymphatic system is a system you may have touched on in anatomy and physiology if you took those courses. And major functions of the lymphatic system are to provide an auxiliary route for the return of extracellular fluid to the circulatory system. It essentially acts as a quote-unquote drain-off system for the inflammatory response. It also renders surveillance, recognition, and protection against foreign materials through a system of lymphocytes, phagocytes, and antibodies. Now, lymph, this is a plasma-like fluid that is carried by the lymphatic circulation. It is made up of water, dissolved salts, and 2 to 5% protein. It transports numerous white blood cells, fats, cellular debris, and infectious agents that have gained access to tissue spaces. Now, the lymphatic vessels will permeate all parts of the body. They are like the cardiovascular system, especially the blood vessels, in the sense that they are integrated into all aspects of the body. The thin walls of the lymphatic vessels are easily permeated by extracellular fluid that has escaped from the circulatory system. Two differences between the bloodstream and lymphatic system. Lymph moves in one direction only. It's not a circular loop from the extremities back to the heart. Lymph is only transported through the contraction of skeletal muscles. There is no heart per se to pump the fluid through the lymphatics. And here you can see the circulatory system versus the lymphatic system, and they very well sort of overlay each other. And here you can see a comparison of generalized circulation of the lymphatic system and blood. Although the lymphatic vessels parallel that of regular circulation, they transport in one direction only, as you can see here, back up to the heart from the extremities. Now, there are several lymphoid organs. Organs and tissues that perform lymphoid functions will include the thymus, the lymph nodes, the spleen, salt, malt, and galt. We'll define all those in a little bit. A loose connective tissue framework that will also house aggregations of lymphocytes, which are called, which can form little nodules, a little bit smaller than lymph nodes. So let's talk about the thymus. The thymus is going to be the site of T cell maturation. Now remember, T cells are part of the adaptive immune system. It is a triangular structure in the pharyngeal region, in the throat. It's largest proportionally at birth. It exhibits high rates of growth and activity and growth until puberty, and it shrinks gradually through adulthood. The reason why it starts to shrink after puberty is at that age, you have been exposed to a bulk of the foreign antigens that you'll be exposed to. And you're not going to see as many quote unquote new things in the numbers as you were from, early developmental years all the way up through puberty when it's most active. Now, the lymph nodes, these are small encapsulated bean-shaped organs. They're stationed in clusters along the lymphatic channels and large blood vessels. Major aggregations of lymph nodes are found in the loose connective tissue in the armpit, the axillary region, the groin, and the neck. The lymph nodes are ideal for filtering out materials that have entered the lymph and to provide appropriate cells for immune reaction. The spleen, this is a lymphoid organ in the upper left portion of the abdominal cavity. It serves as a filter for blood instead of lymph. The primary function of the spleen is to remove worn out red blood cells from circulation. Its immunological function is to filter pathogens from the blood for phagocytosis by macrophages. Associated lymphoid tissues, these are bundles of lymphocytes beneath the skin and mucosal surfaces all over the body. This includes your tonsils, breast of pregnant and lactating women, galt, malt, salt, and balt, and Peyer's patches. Galt, malt, salt, and balt, these are all what we call associated lymphoid tissues. These are essentially scattered throughout the body, all over the place. For example, MALT stands for mucosa-associated lymphoid tissue. Now, looking at blood, blood cells are suspended in a liquid called plasma, which is a clear yellowish fluid. And serum, this is the same as plasma, except it is the clear fluid from clotted blood. And this is what's typically used in immune testing and therapy. The process of the production of blood cells, which would include both red blood cells and white blood cells, is called hematopoiesis. Stem cells, which are the precursors to blood cells, you have a pool of undifferentiated cells These are maintained within the bone marrow. They are immature and unspecialized cells that develop into specialized form and function into mature cells. Now, we really don't care in microbiology, no offense to them, about red blood cells, but we do care about white blood cells because white blood cells are essentially what makes up your immune system. White blood cells are called leukocytes. They are evaluated by reactions to hematological stains that contain a mixture of dyes. Cells are differentiated by color and morphology. They appear with or without color granules in the cytoplasm. And there are two types. We have our granulocytes. These are they contain dark staining granules and lobe nuclei. The agranulocytes, they do not have these dark staining granules and they have very large nuclei. Your granulocytes will include your neutrophils, eosinophils, and basophils. They have prominent cytoplasmic granules that will either stain with eosin or methylene blue. Granules can be released, or both of those dyes, by the way. Granules can be released to kill foreign cells or affect host tissues. Your agranulocytes, they lack these cytoplasmic granules, and there are two types. We have our lymphocytes, and lymphocytes will include your B cells and your T cells, and then you have your monocytes, which will mature into macrophages. And here you can see the blood cells and blood cell lines. Again, we are only concerned with the leukocytes, or the white blood cells. Here's your agranulocytes, which will include the monocytes. Monocytes will mature into macrophages, which are very large phagocytic cells. They have a very high capacity for killing microbes and cleaning up dead cells. And then monocytes can also differentiate into dendritic cells, which will reside in tissues and process foreign matter and present it to the lymphocytes. The second type of agranulocytes are your lymphocytes, and these are your T cells and your B cells. B cells will differentiate and produce antibodies. T cells, this is part of cell-mediated immunity, which will assist B cells. You also have what are called natural killer cells and gamma and delta T cells. Your granulocytes, these are your neutrophils, basophils, and eosinophils, and as well as a fourth one called mast cells. Neutrophils, these are very short-lived phagocytes, phagocytic cells in the blood. They typically show up to sites of infection first. They are active engulfers and killers of bacteria. Basophils will function in inflammatory events, and eosinophils are active in that of parasitic infections such as protozoa or helminths. Mast cells, these are also going to be very active in the inflammatory response, and they oftentimes are what are elevated when we talk about allergies. And then not to discredit the importance of the other blood cells that we find in blood, red blood cells and platelets. We just don't care about these two in microbiology. So cytokines, these are products of monocytes, which will form either macrophages or dendritic cells. Macrophages, lymphocytes, fibroblasts, mast cells, platelets, and endothelial cells of blood vessels. There are four broad categories of cytokines. We have pro-inflammatory cytokines. These encourage specific and non-specific immune responses. Anti-inflammatory cytokines. This will discourage specific and non-specific immune responses. Vasodilators and vasoconstrictors. They change the diameter of the blood vessel or vessel permeability. And growth factor cytokines, which will regulate lymphocyte growth and/or activation. So the mononuclear phagocyte system, this is a support network of connective tissue fibers. It's part of the reticular system, which interconnects nearby cells. It meshes with the massive connective tissue network that surrounds all of the organs. And you can see here you have various tissue cells. And by the way, this is all indicated here in purple. And you can see how you just have these macrophages and dendritic cells just dispersed within the tissues. So monocytes, these will take up residence near the portals of entry or filtration organs. They're waiting to attack foreign intruders. These tissues and specialized histocyte cells include the liver. Resident macrophages in the liver are called Kupffer cells. In the lungs, the resident macrophages are simply called alveolar macrophages. In the skin, the resident macrophages in the skin are called Langerhans cells. In the brain, the resident macrophages are called microglial cells. And again, monocytes, these are phagocytic cells. So let's talk about the physical and chemical defense barriers that we have. The first physical barrier is going to be the skin. Epithelial cells compacted, cemented together, and impregnated with protein called keratin. It is a very thick, tough layer that is highly impervious, and it is also waterproofed. Few pathogens can penetrate this unbroken layer. Really, the only things that really get past the skin is any break that you might have within the a break within the skin. Other barriers that are associated with the skin, we have our hair follicles. The hair shaft periodically gets extruded. Follicle cells are disquamated. And sweat glands, you get the flushing effects of sweat glands that help to remove microbes. Mucous membranes of the digestive, urinary, and reproductive tracts and of the eye. They are moist and permeable. They provide barrier protection without keratinized layer. And the mucus coat impedes the entry and attachment of bacteria. Blinking and tear production will rid the eye of irritants, and flow saliva carries microbes to harsh conditions within the stomach. The respiratory tract, you have nasal hair traps larger particles. Copious flow of mucus and fluids during allergies and colds, it will exert a flushing action. The respiratory tree, this is ciliated epithelium, will move foreign particles entrapped in mucus towards the pharynx for removal. The genitourinary tract protection is through the continuous trickle of urine through the ureters and bladder, which creates a flushing action. Vaginal secretions also provide cleansing of the lower reproductive tracts in females. You also have your microbiome. This provides microbial antagonism. Microbial antagonism is a fancy word for competition amongst microbes. If you have what we refer to as good bacteria living on your surfaces, they are requiring nutrients in order to survive. And there is only so many nutrients available. And if you have a bad bacteria that sort of wants to take up residence, they are competing with those good bacteria. And a lot of times the good bacteria might actually block access of pathogens to the epithelial surfaces. They also create unfavorable environments for pathogens, meaning that they are competing for nutrients, and they can alter local pHs. Non-specific chemical defenses in the skin and mucosal membranes, you get sebaceous secretions, which will exert an antimicrobial effect. Lysozyme is an enzyme that gets secreted by your own cells. It is found in tears and saliva. It hydrolyzes the cell walls of bacteria. That peptidoglycan, it breaks it down. Lactic acid and electrolyte concentrations found in sweat are not conducive for most bacteria to grow on. And the skin's acidic pH and fatty acid content also is not very conducive for a lot of microbes to grow on. With the stomach, you have the secretion of hydrochloric acid. Again, not a very favorable environment for microbial growth. In the intestines, you can have digestive juices and bile. Again, not creating a very favorable environment for growth. Semen has antimicrobial chemicals that are present, and the vaginal tract has a protective acidic pH, much like the skin. Vaginal pH can be as low as 4 to 4.5, which is created by the normal bacteria that live within this vaginal tract. Now, phagocytosis is a process of engulfing cell debris and or pathogens for degradation. The general activities of phagocytosis is to survey the tissue compartments and discover microbes, particulate matter, and injured or dead cells. Phagocytes will ingest and eliminate these materials. They will also recognize immunogenic information, such as antigens, in the foreign matter. Neutrophils, these are white blood cells that react early in inflammatory response to bacteria, foreign materials, and damaged tissue. A common sign of a bacterial infection is you will actually see in a white blood cell count a very high neutrophil count in the blood. Your eosinophils, these are attracted to sites of parasitic infection. They play a minor role in phagocytic antibody-antigen reactions. Your monocytes and macrophages. Monocytes are transformed into macrophages after emigrating out of the bloodstream into the tissues due to chemical stimuli. They increase in size. They enhance development of lysosomes. So macrophages contain very well-developed lysosomes. If you remember, that organelle is of very importance for digestive properties. Not only are macrophages dynamic scavengers, but they also process foreign substances and they prepare them for reactions with B and T lymphocytes. And the stages of phagocytosis are, the first step that has to occur is you actually have to have chemotaxis by the phagocyte. Okay, what chemotaxis means is you actually have to bind to surface markers. If you look here, this little tan thing, these are the receptors, okay, and you can see these little, what they call PAMPs. PAMPs stand for pathogen-associated molecular patterns. We defined that earlier. The molecular patterns are either carbohydrates or proteins on the surface, so they get recognized. That's the first thing that has to happen. Once that recognition occurs, or the adhesion of the bacteria occurs to the surface, you'll get engulfment into the phagocytic vacuole. This will come in through an endocytic pathway, which will form a phagosome. The phagosome will then merge with a lysosome, forming a phagolysosome. Once you form the phagolysosome, the enzymes such as lysozyme, DNase, RNase, proteases, DNase breaks down DNA, RNase breaks down RNA, protease and peroxidases break down proteins and fatty acids, respectively, and lysozyme breaks down the cell walls. So this is going to degrade a lot of those things. Once you have broken down all those things, you'll release the residual debris out of the cell via exocytosis. So pathogen-associated molecular patterns, or PAMPs, they are recognized by phagocytes and other defensive cells. They serve as signal molecules on the surface of microbes, and they're not present in mammals. Examples of PAMPs could include the peptidoglycan itself, LPS, or lipopolysaccharide, which is found on gram-negative bacteria, and double-stranded RNA, which is found in viruses. The pathogen recognition receptors, these are found on surfaces of phagocytes, dendritic cells, endothelial cells, and lymphocytes. They recognize and bind to the PAMPs. They are found on the surface at all times, regardless of whether they have encountered a PAMP or not. Toll-like receptors, this is a term that's used to describe a type of pathogen recognition receptors. They recognize PAMPs, and they set in motion a cascade of events inside the host cell that will amplify and orchestrate a defense response to the pathogen. The phagolysosomal formation and killing, lysosomes will migrate to the scene of the phagosome. They will fuse with it and form a phagolysosome. Granules containing antimicrobial chemicals are released into the phagolysosome that destroy the ingested material. This causes death of the bacteria within 30 minutes. Okay. 4 classic signs of symptoms of the inflammatory response. In addition to phagocytosis, inflammatory response is another immune response. And there are 4 things. We have rubor, This is redness that's caused by increased circulation and vasodilation in the injured tissues. Calor, this is the warmth from the increased flow of blood. Tumor, this is swelling from the increased fluid that has escaped into the tissues. And dolor, this is the pain that's caused by stimulation of the nerve endings. A fifth sign of inflammation is loss of function of that tissue. All signs of inflammation serve as a warning that injury has taken place. It has set in motion responses that save the body from further damage or injury. Functions of inflammation. The chief functions of inflammation is to mobilize and attract immune components to the site of injury, to set in motion mechanisms to tissue repair damage and localize and clear away harmful substances, and to destroy microbes and block their further invasion. And here we can see the major events that occur in inflammation. First, you have your injury and immediate reactions. When you damage the cells or damage tissue, they actually release chemicals that will cause vascular reactions such as vasodilation, which will increase blood flow to the area. The increased blood flow to the area will cause edema and pus formation. And then that will bring lots of white blood cells to sort of clean up the debris. And then you'll get resolution and scar formation as a result. Earliest changes occur in the vasculature during inflammation. The arterioles, capillaries, and venules, you'll see vasodilation. Changes are controlled by nervous stimulation and chemical mediators. These are released by the blood cells, tissue cells, and platelets. And some of them encourage immune reactivity. Some of them discourage or dampen that response. Some act on the blood vessels in the area, and some regulate the growth or activation of white blood cells. Vasoactive substances that cause the endothelial cells in the postcapillary venules to contract and form gaps. This will form exudate, which is blood-borne components that escape into the extracellular space, and edema, which is local swelling and firmness due to the accumulation of the exudate in the tissues. Movement of the white blood cells from the bloodstream into the tissues occurs because white blood cells are actively motile and they're able to change shape. Receptors on the endothelial cells will capture the white blood cells and participate in their transport out. And that process is called diapedesis, words right here, where the white blood cells are attracted to the chemicals outside the blood vessel. So they migrate to the walls here and then they squeeze their way out. The tendency of white blood cells to migrate in response to a specific chemical stimulus given off at a site of injury or infection is essentially due to chemotaxis. Cells swarm from the body compartments to the site of infection and remain there to perform general and specific immune functions. The influx of fluid will dilute toxic substances. The fibrin clot can trap microbes and prevent further spread. Neutrophils will actively phagocytose and destroy bacteria, dead tissue, and particulate matter. Pus is actually whitest mast cells, liquefied cellular debris, and bacteria. The late reactions of inflammation, you have long-lived inflammatory reactions, which will attract monocytes, lymphocytes, and macrophages. The macrophages will start to clear the pus, the cellular debris, the dead neutrophils, and the damaged tissue. The B lymphocytes will react with the foreign molecules and start to produce antibody production. The T lymphocytes will kill intruders directly, and the tissue is completely repaired and replaced by scar tissue. Now let's talk about another type of inflammatory response, which is a systemic inflammatory response. A fever, this is an adjunct to inflammation. A fever is an abnormally elevated body temperature, nearly universal symptom a symptom of an infection. It's also associated with certain allergies, cancers, and other organic illnesses. The ranges of a fever, you have a low-grade fever, which is between 100 and 101 degrees Fahrenheit. Moderate's 102 to 103, and a high fever is 104 or higher. High fevers need to be addressed as they can lead to death if they're not brought under control. A bunch of chemicals called pyrogens will reset the hypothalamic thermostat. Your body's thermostat is found within the hypothalamus to a higher setting. Exogenous, this is originating outside the body, which would be products of infectious agents such as viruses, bacteria, protozoan, and function, or the release of endotoxins can cause pyrogens to be released. Endogenous, this is originating inside the body. This is released by monocytes, neutrophils, macrophages during the process of phagocytosis, which could also cause a fever to be produced. Now, the benefits of a fever is that it inhibits multiplication of temperature-sensitive microorganisms. It impedes the nutrition of bacteria by reducing the availability of iron, and it increases the metabolism and stimulates immune reactions and naturally protective physiological processes. Treatment for fever. This is a difficult decision to suppress a fever or not. Some advocates, slight to moderate fever in otherwise healthy person should be allowed to run its course, as there are many potential benefits with minimal side effects. All medical experts do agree that high and prolonged fevers are risky and should be treated. Fevers in those with cardiovascular disease, head trauma, seizures, and respiratory ailments should also be treated. Antimicrobial proteins or antimicrobial substances. These are proteins that are produced by your cells that have antimicrobial natures. One class is interferon. This is a small protein produced naturally by certain white blood cells and tissue cells. It's used in therapy against certain viral infections and cancer, and it's involved in the defenses against viruses and other microbes and immune regulation and intercommunication. Interferon is not virus-specific. Synthesis in response to one cell type will also protect it against other cell types. It's produced industrially for the treatment of virus infections. Complement, this consists of over 30 blood proteins. They work together to destroy bacteria and viruses. Complement oftentimes will set up what we call cascade reactions, which is the sequential physiological response, which will lead to the death of those organisms. And there are three types of complement pathways. We have the classical complement pathway, which is activated by antibodies, which are bound to microbial surfaces. The lectin pathways, which is activated by lectins, which are surface proteins that can be found on bacteria. An alternative pathway is activated directly by repeating molecules on the microbial surfaces, such as LPS. Again, LPS is something that can be found on gram-negative bacteria. And then we have, besides complement and interferon, we have what are just called antimicrobial peptides / proteins. These are short proteins that are capable of inserting themselves into bacterial membranes. They're typically between 12 and 50 amino acids in size. These include things called bacteriocins, defensins, protegrins, and they are all part of the innate immune system. They have an effect on other actions of non-specific and specific immunity. So again, this chapter covered both our first and second lines of defenses. Again, your first line of defense was all of your barriers, such as your skin, mucosal membranes, microbiome, so on and so forth. Second line of defense included things like phagocytic cells, inflammatory responses, fever, antimicrobial peptides, proteins, all those things along those nature.